So today we will talk about life cycle of HIV. So HIV stands for Human Immunodeficiency Virus. So I have talked about the structure of HIV in my previous video. So let me recall that structure, right? So here this is the capsid and there are two single double-stranded RNA in this. Then we are having this matrix protein over here. Then we are having double membrane. This is enveloped, right? So this is this over here. Then there are glycoprotein GP. You can see over here. So over here this is GP120, this is GP41, this is P24, this matrix protein is P17, right? So I have talked about this structure in my previous video. So now let's talk about the life cycle of HIV. So the first stage which we are having is, obviously it will be attachment, right? It will be attachment. So attachment. So what happens over here? In this step, virus attaches to the surface of T helper cells. So as you talked about that the target cell of HIV are T helper cells, macrophages, and some brain cells too. So now it will attach. For example, this is a T helper. Consider this is a T helper, and it will get, and consider this is the this is the virus, and this is the brain. If you will be wondering that why it is attacking on T helper, because the T helper contains a receptor known as CD4. Okay, what does CD4 means? CD4 means that cluster of differentiation. Cluster of differentiation. So consider this is the HIV virus, and it will get attached to this. For example, let me make this over here. So over here this will attach to this. Now this GP, this is GP120, right? This GP120 will identify the CD4, like this GP120 will identify the CD4, and then it will get attached to this T helper. What happens that it get attached, and this GP41, this helps in the second stage, which is fusion. So over here what happens, this GP120 reads this CD4. It actually identifies this in order to get attached. So here it attaches. The GP120 get attached to the CD4 over here. Now let's talk about the second step, which is fusion, right? So what happens over here is that, as you can see over here, the CD4 is, the GP120 has read CD4, has identified it. Now what will happen, this T helper cell, right? Consider this is a this is T helper, right? Now this HIV virus this will enter into this now. this will enter. It helps in the fusion of the viral envelope with the host cell membrane. When it fused, shades its protective coat and enter the RNA along the enzyme. Now what will happen? This fusion, this GP41 enters the cell and it will insert its viral RNA into this. Right? It will insert its viral RNA into it. Let me show you the diagram too. So as you can see in this, okay, so as you can see in this diagram over here, this is right. Now what's happening, this is the RNA, this is the double-stranded RNA is going inside, right? So the attachment and the fusion has been done over here, and now the RNA will go inside the cell. RNA will go inside the cell through this GP41. It will enter and it will shed its protein coat outside the host cell, right? The protein coat will not go inside, right? Remember this thing, that protein coat will remain outside, right? And the RNA, double-stranded RNA, will move to the host cell, to T helper, by this GP41. Now let's talk about the third stage, which is reverse transcription. Third is reverse transcription, right? Reverse transcription. Okay, HIV virus has 3 enzymes, right? It will take these enzymes along, like these enzymes will also enter the host cell along with the genome, the double-strand RNA, which are 3 enzymes: protease, integrase, and reverse transcriptase. So here reverse transcriptase will work. Reverse transcriptase will work. What is this now? Reverse transcriptase is actually a enzyme, right, that will do reverse transcription. Now what is actually transcription, right? Let's talk about the simple transcription. What is transcription first? Transcription means conversion of DNA into RNA, right? Transcription means conversion of DNA into RNA. But what is reverse transcription, right? So let's talk. What is reverse transcription, right? The reverse transcription is the conversion of RNA. It is the conversion of this RNA into DNA. So I hope you understand the concept of transcription and reverse transcription. What is transcription? Transcription is actually the conversion of DNA into RNA, and reverse transcription is the conversion of RNA into DNA. So as you can see, what is the genome of HIV? The genome of HIV is double-stranded RNA, right? There are 2 strands of RNA over here, right? So here this RNA will be converted into cDNA. Now what is cDNA? By the help of reverse transcriptase, by the help of this enzyme, by the help of the enzyme reverse transcriptase, RNA will be converted into cDNA. Now what is this cDNA? You will be wondering. cDNA means let me write over here. cDNA means complementary DNA. cDNA means complementary DNA. Consider, let me draw a diagram. Over here we are having the host cell, right? Over here this is the nucleus, right? So in this was double-stranded RNA. Now this is RNA, right? And now it has been converted into DNA. This is cDNA. It's converted into cDNA, and still one strand is remaining. Now what will happen? RNAs. There is another enzyme. This enzyme is of host cell. So basically HIV uses its own viruses, which are protease, integrase, and reverse transcriptase. But here the enzyme uses RNAs, right? Here the enzyme used is RNAs, right? This is host cells enzyme. Host cell enzyme. This is not HIV's enzyme. This is a host cell enzyme. What will this RNAs do? It will destroy, or it will disintegrate this one RNA, right? Now what will happen? This RNAs will destroy the, or you can say it will disintegrate the RNA. It will disintegrate the RNA. As I told you before, we were having over here RNA. Now that has been converted into cDNA, but still one strand is left, and that will be disintegrated by the RNAs. Now what will happen? Okay, I've combined 4 steps over here. Okay, what will happen actually? Reverse transcription. The work of transcription has been done. Now from here the 4th step have been started, which is integration, right? Over here 4th step have been started, integration. What will happen? Integration. RNAs, which is host cells enzyme, will come, and this will disintegrate the RNA of the HIV. Now it will disintegrate the RNA of HIV. Then what will happen? Now the cDNA will be converted into dsDNA. It will be converted into, in integration, the cDNA, which is complementary DNA, it will be converted into double-stranded DNA. What is cDNA, which means complementary DNA? DS means double-stranded DNA. cDNA will be converted into double-stranded DNA, and then in the step of integration, this double-stranded DNA will get integrated. It will join the genome of the host cell. The host cell DNA. It will join host cell DNA by the help of enzyme known as integrase. And this is HIV's own enzyme. This is enzyme, right? The double-stranded DNA and the host cell DNA will combine together by the help of integrase enzyme, and this process is known as integration, and it will form provirus. Now that condition will known as provirus, and now provirus is actually will be gone into the latent period. Now that provirus virus will move to latent. Now this provirus will move to latent period, or you can say this is latent period means dormant period. Move to dormant period. Okay, why it is moving this? What's the reason that why the provirus is moving to latent period? Why it's going in dormant period? Because it has, like, research is still going on, that maybe because of the effects of reverse transcription. So maybe because the effects of reverse transcription, the provirus is going into latent period or dormant period. Now let's summarize what we have studied till yet. So the first step was attachment. The GP120 will identify the CD4, right, and it will get attached to this. Now in fusion, this GP41 will move inside the host cell, and the RNA will be transferred into the host cell. Third is reverse transcription, in which the enzyme reverse transcriptase will work. So in transcription, DNA is converted into RNA, but in reverse transcription, RNA will be converted into DNA. Now RNA is converted into cDNA, which is complemented DNA by the help of reverse transcriptase, right? Yes, and here the step reverse transcription is completed. Now we are having the fourth step, in which, like, we know that RNA has been converted into cDNA, but still one strand of RNA is remaining, that will be disintegrated by the help of enzyme known as RNase, which is the host cells enzyme, not HIV's virus enzyme. It is host cells enzyme. Now the cDNA will be converted into double-stranded DNA, and then this DNA, double-stranded DNA, will join the host cell DNA to form provirus by the help of enzyme known as integrase, which is the enzyme of the HIV, right? HIV has brought this enzyme known as integrase, and this whole process is known as integration, in which double-stranded DNA joins to host cell DNA and will form provirus. And provirus often goes into the latent period, dormant period. Why does it go in dormant period, in latent period? Because of the effects of reverse transcription. Okay, now let us discuss the fifth step, which is replication, right? Now we know that double-stranded DNA like provirus has been formed, right? Now that DNA will form mRNA. That DNA will form mRNA by the help of enzyme RNA polymerase. By the help of enzyme RNA polymerase. This is a host cell enzyme. What happened? Replication, like, obviously now transcription will occur, so now DNA will be converted into mRNA. Now this mRNA will go into protein, now this mRNA will go into the ribosome. Okay, before talking to this, now two types of mRNA will be formed. What will be that two types? One mRNA will go to ribosome. One will go into the ribosome, and one mRNA will go to the cytoplasm, right? One will go into cytoplasm. For what reason? For the production of other HIV viruses, for the replication of other HIV viruses. Obviously RNA is required for the production of HIV virus. So this the mRNA formed, there will be two types of mRNA formed. One will go into the ribosome and one will be going to cytoplasm, and the one which goes in cytoplasm is known as genomic RNA, that is known as genomic mRNA, right? And the one which goes in ribosome, like why it will go in ribosome? Obviously for translation. What is translation? Now formation of our mRNA to the protein is known as translation, right? And this mRNA will move to ribosome and they will form protein. They will form protein by the help of translation, right? Now the mRNA will, there will be two types of mRNA. First genomic mRNA and the normal one mRNA, which will go in ribosome and then it will form protein by the help of translation. Now we are having the sixth step, which is assembly. Now what happens, like after the translation, in the result of translation and in ribosome, the proteins are formed, right? There are larger subunits of protein have been formed, right? What is now translation? You will be wondering this. Let me tell you. Translation is the conversion of RNA, or you can say simply you can say is the conversion of mRNA into protein is known as translation. Now there will be larger units of protein, larger there will be your larger units of protein. Now these larger units of protein will be cut into smaller units. This will be converted into smaller units by the help of enzyme protease. By the help of enzyme protease. By the help of enzyme. Now it is not host cell's enzyme. This enzyme was brought by HIV. It was brought by its own, right? So now this larger units will be converted into smaller units by the help of protease enzyme, where these subunits are assembled to form wire structure. Why these smaller units of protein are required? Smaller units of what? Smaller units of protein are required. Units of protein. Why it is required? In order to form virus structure, this protein is required smaller units of protein. Obviously the matrix protein, the capsid is made of protein, right? So obviously they need smaller units of protein. For that purpose the protease enzyme is used. Now what will happen, the last step, which is budding. What will happen over here is seventh step, which is budding, right? Here you can see this is budding. What will happen in this when complete? Okay, now we know that the RNA has been formed for the genome, the proteins have been formed. Now virus, like a virus will be made. Whole structure is completed. Is completed over here, right? Structure is completed over here, and complete virion. I know, like I hope that you know the concept of virion. Okay, complete virion is formed. Now what will happen, that virus will be released through budding. Now virus will be released by the host cell. Virus will be released released through which process? Through budding. Okay, have you seen the potato budding? Buds are over there. There are that are full of stars, that are full of carbohydrates, like that over here. There was the same process. You can wonder how it will happen. What will release through process of budding over here. Now, because, and what, the cell membrane of that host cell get attached to that virion. That's why, that's the reason behind HIV virus is the enveloped virus, because there is a double protein layer, lipoprotein is there. Why it is there? Because it gets the cell membrane of the host cell. That's why it's enveloped. Now let me show you the picture so you can summarize it easily. Okay, now see in this picture, right? Now this was the virus, the CD4 and the GP120 identified each other. This is the area where fusion is happening, right? This is the second step. Now here, like, don't look at these steps, look at the steps which I am telling you, right? Now the reverse transcription will occur. Obviously the RNA will be converted into cDNA, which is complementary DNA, right? This is a reverse transcription. Now in integration, the cDNA will be converted into double-stranded DNA, and the one strand of RNA will be disintegrated by RNase, which is the host cell's enzyme. Now this double-stranded DNA will do transcription. This double-stranded DNA will form mRNA, right? And now there are two types of mRNA, genomic mRNA and the normal one. The genomic one mRNA will go in cytoplasm for the production of the more HIV genome, and the normal will go to the ribosome for the process of translation. What is translation? Translation is just a process in which the mRNA is converted into protein. Now in that stage is known as replication, in which mRNA is converted into protein, right? Now in assembly, what happened, that the larger unit of protein are cut into, are broken down to smaller units by the help of enzyme protease, right? What is protease? Protease is an enzyme which is used by host cell. Now what will happen when it will cut down to smaller units in the seventh step, which is budding? You can see over here and here, this is budding. This is known as budding, like how it is coming out from the cell. This is known as budding, right? I hope the concept is clear to you. And I forgot to mention in that's double-stranded DNA, and then double-stranded DNA will join together to host cell DNA by help of enzyme integrase, and it will form provirus, and then provirus will go in the latent period or the dormant period, right? Let me show you another example of this. Here it is. You can see, easily understand this. From here, that fusion is happening, right? This is the viral RNA. RNA is converted into DNA, right? This is a reverse transcription. Now after that, it is joining to the host cell's DNA. Now it is forming a provirus. Then after provirus, there is a process of replication, then assembly, and then budding. This process is known as budding. And you can obviously see that there is a cell membrane. This cell membrane is of this host, and now ultimately host cell will get destroyed. So I hope the concept of life cycle of HIV virus is clear to you. If you are having any query, you can comment me and I will be happy to answer.